everyone. In this lesson, we're going to discuss cell structure, which essentially means we're going to discuss what cells are and what their function is. The opening sentence to any introductory lessons about cells always includes the statement that cells are the basic structural and functional unit of life. But what does that even mean? The term structural unit of life means that each cell has a unique structure which behaves as a fluid sac inside which all the chemical reactions of life are occurring. And these reactions are occurring independently regardless of whatever is happening outside of that cell. So the basic structure of life, or the basic structural unit of life, implies that this structure is the smallest structure inside which life can exist. The functional part of this statement means that it is these structures, or these are the smallest structures inside which the function of life can occur. Function of life means unique chemical reactions that are necessary to keep the organism alive and living. And cells serve as the smallest unit inside which all of these reactions can occur that can help sustain life. And so, the smallest structure inside which a life can exist is a cell, which is why they're called the basic structural and function unit of life that are arising from pre-existing cells. These cells can behave as the building blocks of life because many such cells can come together to give rise to different types of tissues. So each cell will be performing a different function, and cells that are performing the same function can come together to form a single type of tissue. Many different types of tissues can then come together to form a single organ. Many organs come together to form organ systems, which finally gives rise to a whole organism. And so, without a cell, there will be no tissues and hence no organs or organ systems, which is why these simple cells are considered the building blocks of life, which are responsible for making up the whole organism. But cells are not empty structures and instead house various organelles such as the nucleus and the mitochondrion that help the cell carry out essential processes of life, which helps the organism stay alive. Organelles are structurally and functionally distinct part of a cell, which may be either surrounded by a single membrane, a double membrane, or no membrane at all. They basically allow for compartmentalization to occur inside the cell so that each organelle is working independently of the other organelle. The term organelle is actually derived from the term organ, since each organ within our bodies is responsible for doing a different or specific function for our entire body. Similarly, each organelle is responsible for carrying out its unique and characteristic function for the cell. So, these structures found within the cell behave as tiny little organs for the cell they are present in or found in. And each one of these organelles has a unique structure and function. Now, no two cell types found within the body are the same. So, a cell that is making the tissue of, let's say, your stomach is going to be a very different kind of cell versus the cell that is responsible for making your heart. Despite those differences, there will be some organelles that are going to be characteristics of all living cells. And then there will be some organelles that will be found in both animal as well as plant cells both, but some of them will have some organelles that are either only found in animal cells but will not be found in plant cells, but then there will be some organelles that can only be located inside a plant cell and never inside an animal cell. For this lesson, we will be focusing on the cell membrane or plasma membrane. We'll discuss what a cytoplasm is, and we'll discuss a detailed structure of the nucleus. And if you've been studying biology over the past few years, I'm sure you all are already aware of these organelles or structures found within the cell. But in this lesson, we will be discussing them in far more detail than you have already studied. Remember at the start of this lesson, I said that within each cell, there are different chemical reactions happening that helps this particular cell to sustain life. For any place to have a reaction, it initially needs a certain amount of raw material, and then that raw material is used to make a product. If the cell was fully permeable, whatever was entering the cell would instantly leave, also depending on whether the concentration of that substance was higher inside the cell or outside the cell. And so, to prevent this from happening, there had to be some sort of boundary or lining that would limit what enters the cell and what leaves the cell, so that a concentration gradient could be maintained between the inside of the cell and outside of the cell. This boundary was called a cell membrane, and cell membranes were known to be partially permeable structures, which essentially meant that these structures would only allow certain substances to enter the cell and similarly would only allow certain substances to leave the cell. They would not randomly allow every single substance to enter the cell or leave the cell. So, if something was trying to enter the cell from outside the cell, the first structure that they would encounter that would limit its entry would be a cell membrane. Similarly, if any product was trying to leave the cell, it would had to cross a membrane in order to leave that organelle or cell. Now, I'd like to highlight that in this lesson, when we say membrane, we would particularly be talking about a plasma membrane or a cell membrane, which is the membrane that surrounds the entire cell. But the nature of the membrane is going to be similar whether that membrane is a plasma membrane or whether that membrane is a membrane that surrounds an organelle. Since I did point earlier that an organelle can either be bounded by a single membrane, a double membrane, or no membrane at all. So, in case it is bounded by a membrane, the properties of that membrane will be very similar to the properties of the cell membrane. And we do have an entire chapter dedicated to membrane, so we won't be going in a lot of details about it. But for this lesson, we're only going to talk about very briefly what their structure is and what their function is within a cell. So, to start off, membranes are considered partially permeable structures because these membranes are made of something called a phospholipid. What phospholipid is will be covered in our chapter on biological molecules, but at this point, just keep in mind that phospholipid is an example of a lipid substance. And lipids generally are considered nonpolar in nature, which means that they repel water. But phospholipids are slightly different from a typical lipid molecule, such that it has a phosphate head which is considered polar, whereas the central part of it is called the phospholipid tail is nonpolar in nature, so that repels the water. Now, the cell is unique in the sense that the intracellular space, which means the inside of the cell, has an aqueous environment. Similarly, right outside the cell, which is known as the extracellular space, is also an aqueous environment. So, this is aqueous and this is aqueous. This, on the other hand, has to serve as a barrier between this space and this space. If this barrier did not exist, whatever was happening here would simply diffuse here because this environment and this environment were of the same nature. They were both aqueous. But since now this layer is present between these two spaces, this middle part of this layer is going to repel water, which is why it's going to be centrally located, and this part, which is the phosphate head of this phospholipid molecule, is going to be attracted towards water, which is why this part is going to face the water and this part is going to face the water. So this part can be located towards the outer end of the cell, and this head can be located towards the inner side of the cell membrane, which is why both these phosphate heads allow this structure to stay in contact with both the environments, which is why whatever is happening inside can still stay in contact with whatever is happening outside with the help of this membrane. But this tail present within the phospholipid molecule is going to repel the water on both sides, which is why it will not allow the water here or the aqueous environment to freely mix with the aqueous environment outside. And just to highlight, since at this point you're not going to be aware of the structure of phospholipid, this one structure is an example of phospholipid. So this is the phosphate head of the phospholipid molecule, and this is the phospholipid tail. So this is a single molecule of phospholipid. Since this is one layer of phospholipid molecule and this is the second layer of phospholipid molecules, this entire layer together is called a phospholipid bilayer. Now, if you notice this diagram as well as this diagram, you can observe that from the cell membrane, there are these finger-like projections that are being protruded or coming out from the membrane itself. These finger-like projections of the cell membrane are called microvilli, and their sole purpose is to increase the surface area to volume ratio of the cell membrane. What that essentially means is that over this area, if the membrane was a single layered membrane, this is the only membrane that would be covering this area. But now within this area, if you notice, it's all of this that's a membrane. So now within this same area, there is an amount of membrane that is at least thrice or quadruple times more than the actual area of the original cell membrane had it not been thrown into these folds. Since I mentioned earlier that whatever substance is either entering the cell or leaving the cell has to do that through the cell membrane, that means whatever is entering the cell from the extracellular space into the intracellular space is getting absorbed into the cell, and whatever is being secreted out of the cell, that means whatever is being made inside the cell or the cell needs to get rid of it to the extracellular space from its intracellular space, that substance is secreted out of the cell. So with an increased surface area due to microvilli being present on the surface of the membrane, the rate of absorption and secretion increases in case that was the area at which absorption and secretion was occurring. So now within the same time period, more substances are being absorbed and secreted over the same limited area because the membrane has been thrown into multiple folds, that is increasing the surface area to volume ratio of the membrane. But since exchange of material needs to occur through this membrane needs to be extremely thin. And that's why membranes usually have a size of around 7 nanometers. So membranes are only 7 nanometers thick. And if you recall from our lessons on microscope, you'd remember that the maximum resolution that a light microscope can offer is only 200 nanometers. But since the membrane is only 7 nanometers thick, that makes it far smaller for it to be viewed under a light microscope, which is why a detailed structure of the cell membrane can only be viewed under an electron microscope. And in order to view anything under a microscope, we need to stain it first. And when we do that, the hydrophilic heads or the phosphate heads of the phospholipid molecule take up the stain more darkly, whereas the phospholipid tail that was present between the two layers of the phosphate head actually takes up the stain far more lightly, which is why under a microscope the membrane gives a trilaminar appearance, which can be a little misleading because I've already pointed out that the membrane is a bilayer structure, not a triple layer structure. But because of the stains that we use to stain the specimen to view under the electron microscope, the stain makes it look like that the membrane has 3 layers instead of 2. The phosphate heads on both sides appearing darker, and the phospholipid tails appearing paler in the center. Let us now quickly discuss what a cytoplasm is. So, for example, if a substance was trying to enter the cell from the extracellular space into the intracellular space, the first structure it was encountering was the cell membrane, which was serving as a partially permeable barrier or membrane to that substance. But let's assume that substance has been able to cross this partially permeable membrane. Now, the first location where that substance is going to find itself within the cell is going to be the cytoplasm. Cytoplasm is a watery or jelly-like substance that is found between the cell membrane of the cell and the nucleus of the cell. By composition, the cell cytoplasm or cytosol is about 80 to 90% water, and the remaining part is made by a combination of different kinds of salts and minerals. The consistency of cytoplasm is similar to that of a jelly, and cytoplasm is responsible for housing all the organelles of the cell that are found within the cell membrane of the cell. The cytoplasm of the cell also serves as the site where most of the chemical reactions necessary to sustain life are occurring within the cell. So, cytoplasm has many different kinds of organelles as well as many different kinds of enzymes that are necessary to carry these reactions out. Let us now move on to our next lesson, which is going to be about the nucleus.